All right, we left off part 2 here for gel electrophoresis. We left off talking about this picture right here, which is a picture of the gel, and you can see that it's been stained, and then under ultraviolet ultraviolet light, you can see that the bands of the DNA are showing very nicely. And remember, this is the negative end, this is the positive end. These are our DNA wells. And we have run this gel, then stained it, and now we're taking a look at it. This, for example, is like 200 base pairs all the way down here. I said this one is like 15 base pairs. Doesn't matter. It's in that general range. It doesn't have to be specific on how big the segment is in base pairs. But like I was saying, this can be used for investigating and figuring out the good old whodunit question. So if we say that this one right here is our crime scene data, or DNA, crime scene DNA, and this is suspect number 3, this is suspect number 2, this is suspect number 1, this is suspect number 4, and this is suspect number 5. Now you can see that the crime scene DNA that we have has bands that are here, 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 and then all the way down to there. So we can very quickly see that suspect number 1 has a whole bunch of different segments from the crime scene data. Suspect number 2 doesn't have any of those bands that match. Suspect number 3 has the exact same fragments all the way down. And that, of course, would lead us to the DNA evidence of suspect number 3 being at the crime scene due to the matching of the bands of the DNA. Now, these bands were cut by restriction enzymes, or this DNA. These DNA segments were cut by restriction enzymes. And these aren't just one single strand of DNA. This is DNA that has been copied multiple thousands of times. And these are literally thousands of segments of DNA that are piled up on one another at this exact same location because they're all the exact same size. So all these are the exact same size. So this would lead us to suspect number 3 being the person who was at the crime scene, because these are the DNA evidence that we've collected from the crime scene. This could also be used to help figure out families, whether or not they're related, and things like that. So gives you just a little idea of what some of these things can be used for. Those restriction enzymes that I mentioned before are also known as restriction endonucleases, or DNA scissors. They cut in a specific sequence, they cut DNA. And this was originally served or used as a natural defense for bacteria. We learned about this enzyme, stole that enzyme and a few others from bacteria, like we've done with many of our genetic technologies. And it was used against the defense of viruses coming in. The restriction enzymes cut the viral DNA up into pieces so that it wasn't able to go through its normal viral activity, taking over the cell and making copies of itself. If it's in pieces, it can't do that. So the bacteria protect their own DNA from cutting by using something known as methylation, and that's basically methyl groups that protect those segments of DNA. So here are our restriction enzymes, or endonucleases. We have EcoR1, HIND3, BAMH1, and EcoRV. All of them recognize a different sequence. For example, EcoR1, every time it finds a GAATTC, it cuts that sequence, or cuts at that sequence of genetic information. Now, this enzyme doesn't care what is there. It can cut in the middle of a gene, it can cut before a gene, it can cut after a gene. It just finds every single GAATTC in the sequence and cuts. So this is how we get our fragments of DNA. Now, if you look at EcoRV at the bottom here, it cuts at a different sequence. It cuts at GATATC. So the recognition sequence is also palindromic, which means it can be spelled the same way forward or backward, like erase car or kayak. You get the idea. So here comes BAM-H1. It's cruising down the DNA. It finds its segment, GGATCC, and it makes a cut. In this case, it makes a cut in something called sticky ends, because these are unpaired bases here, and they can stick or attach to other bases, such as GATC. Now, this is how we are able to splice things like the insulin gene or the gene for human growth hormone into bacterial plasmids, which you read about in genetic technologies. Those are sticky ends. Another type of enzyme, EcoRV, makes something called blunt ends. Those blunt ends obviously don't attach to anything else, and this is the type of enzyme that we would use, restriction enzyme that we would use to cut up DNA. Because we don't want DNA accidentally sticking back together and giving us false positives. We want those segments on their own. So for example, the first segment or size of base pairs would be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this would be 10 base pairs. And the second segment would be 1, 2, 3, 4, 5, 6, 7. 7 base pairs. All right.